Hello and welcome to this video on the primary structure of protein. According to hierarchical model of protein folding, primary structure is the first level of structural organization in a folded protein. The topic is good for short notes and MCQs for any examination. However, to learn this topic effectively, it is very important that we learn the basic structure of amino acids, associated groups, and how they are connected to form peptide bond. Peptide bond plays an important role in the formation of primary structure, therefore its feature will be discussed also. Then I will discuss the significance of primary structure in learning pathogenesis of human diseases. Then the main topic, features of primary structure, and finally I will discuss how to determine the primary structure of protein. So protein is a polymer of amino acids, therefore I will present the basic structure of amino acid. Each amino acid or every amino acid has an alpha carbon atom. Attached to it are carboxylic group, amino group, and H group. These three groups are same in all amino acids except proline. The difference lies in the R group. R group is unique. There are 22 different types of R groups. Therefore, there are 22 different types of amino acids. These are also called as proteinogenic amino acids because they are used in making protein or in protein synthesis. The amino group and carboxylic groups are important in connecting amino acid through a bond called peptide bonds. Therefore, the peptide bond is the backbone of the protein or the primary structure of. So, peptide bond formation between two amino acids is a condensation reaction between the amino group of one amino acid and carboxylic group of the second amino acid. It leads to the formation of single covalent bond which is known as peptide bond. It is accompanied with the liberation of water molecule. To understand the primary structure of protein or protein folding, it is important that we review the features or characteristics of a peptide bond. If you can see here in this structure, the peptide bond is a single covalent bond. However, electron pair at nitrogen atom causes dislocation of electron cloud, leading to double bond character in the peptide bond. As a result, it becomes rigid, and rigidity does not allow free rotation around peptide bond. So peptide bond is a planar rigid, and it has a plane made up of 6 atoms, 3 carbon, 1 oxygen, 1 nitrogen, 1 hydrogen. And in this structure or in this plane, the carbonyl oxygen and amide hydrogen are in transposition. So these are the features of a peptide bond. So you have seen that the peptide bond has partial double bond character, and as a result of it, it does not allow free rotation, and therefore the angular rotation around peptide bond is restricted. This angle is known as omega angle. However, the plane of peptide bond has freedom of rotation around the single bond between the amide nitrogen and alpha carbon atom. This angle is known as phi angle. Similarly, there is freedom of rotation, angular rotation around the single bond between alpha carbon atom and carbonyl carbon atom, and this angle is known as psi angle. So these three angles play a significant role or very important role in protein folding or protein structure. So now coming to dihedral angles, I will discuss what are dihedral angles, how they are formed, and what is their impact on overall protein structure and folding. So this is a polypeptide chain where I have shown multiple planes of peptide bonds. At each alpha carbon atom, these bonds they intersect, they rotate, forming two types of angles. They are known as phi angle and psi angles. Collectively, they are called dihedral angle or Ramachandran angles or torsion angles. So these are very important factor in determining overall structure of protein and protein folding. So coming to the main topic, the primary structure of protein, I will present the important features of this structure. This is a unique linear arrangement of amino acid. It is unique because there is a gene encoding such structures. Every protein or polypeptide has a unique structure, primary structure, because there is a gene encoding such structures. This is formed by the linking of amino acid through peptide bonds. All amino and carboxylic group present in the amino acids are utilized in the formation of peptide bond, leaving one free amino group at one end and one free carboxylic group at another end. So there are two ends in the molecule. One is called N terminus, having free amino group, and C terminus, or the end, having free carboxylic group. So this is called as polarity in the molecule. This is the way protein is synthesized, protein is written, protein is also sequenced by chemical methods. You will learn that. So this is the primary structure. Now these peptide bonds are pretty stable. They are not broken by mild denaturants such as pH and heating or etc. Heating, when I say, it has relevance to your digestion or meal preparation. When you cook your meal, you boil egg, the protein is simply denatured or unfolded. It is not hydrolyzed into its building block or amino acids. Normally, amino acids are the primary sequence or the primary structure of protein is linear. However, you will see some instances where the primary structure is branched as well. Example is insulin. So regarding the significance of the primary structure, or in other words, why we have to understand or learn primary structure of a protein, right? I would like to explain this in terms of two very important points. Number one, as we know that the primary structure or the structure of a protein dictates its biological function, therefore by knowing primary structure we can understand structure function relationship. Second point is regarding the disease pathogenesis. Primary structure change, there are many diseases which are caused due to change in the primary structure of protein, leading to diseases, right, such as cystic fibrosis, which is caused due to deletion of a single phenylalanine residue from the primary structure of CFTR. Huntington disease or neurodegenerative disease is caused due to addition of multiple glutamine in the primary structure of Huntington protein. Similarly, sickle cell disease is caused due to substitution of glutamate by valine residue in the primary structure of beta globin chain. So how to determine the primary structure of a protein? It is determined by a method which was developed by Sanger, known as Sanger's method of protein sequencing. He sequenced insulin chains and he got Nobel Prize. However, the method I am going to discuss is known as Edman degradation, in which a different chemical is used, which is PITC or phenylisothiocyanate. So here I will discuss Edman degradation. So just to give you an idea how the method is done, this is the flow chart of the sequencing method. The first thing is you have to isolate or purify a polypeptide. If you have more than one polypeptide in your protein, you have to separate them and purify individually. So you have to start with a purified polypeptide. Then you cleave the polypeptide into overlapping fragments using chemical or enzymes and purify these fragments and individually sequence them, and by aligning the derived sequence you can obtain the overall sequence of your protein. So now coming to the Edman degradation sequencing method, basically if you want to sequence, let us say, this peptide, oligopeptide having 6 amino acid residue, the first being alanine. So there are three basic steps in this sequencing method. The first one is labeling of the N-terminal amino acid with Edman reagent, which is PITC or phenyl isothiocyanate. Adduct is formed, and this adduct basically causes change in the immediate peptide bond. This bond becomes acid labile, and therefore the second step is cleavage of the labeled amino acid by acidic pH, and the third step is identification of the cleaved amino acid using HPLC. You can identify the amino acid. This will leave the peptide length n-1. So if you want to sequence completely, you have to repeat these three steps in a cycle till the last amino acid. So in summary, protein sequencing involves three steps. The first one being the labeling of N-terminal amino acid using PITC. Second is cleavage of the labeled amino acid using acidic pH. And the third or final step is the identification of the cleaved amino acid using HPLC. So these three steps are repeated again and again to obtain the full sequence of a given protein. If you think this topic is helpful for you to understand protein structure, please like it, subscribe it, and share with your friends who are interested in protein biochemistry. Thank you and take care. Bye bye.